Data, what kind of analysis are we going to perform as a biologist, and what kind of questions we are going to ask? If we talk about this, we can classify it into two categories, clustering and classification. For clustering problems, look for questions like this. Which genes have similar function? In other words, which genes show a similar behavior under different environmental conditions. For example, if these genes are in a pathway, in a biological pathway, and if this pathway is active under certain conditions, all genes in the pathway are going to be expressed in order to activate that pathway. So genes usually do not perform their functions alone. So genes usually are in harmony with other proteins. They are part of larger workflows, larger pathways to perform certain functions. So, and having the cell is usually really efficient. The cell does not want to have one gene in that pathway expressed just for nothing. Okay, I mean every protein has a cost. And therefore, in order to activate that pathway or in order to deactivate that pathway, either you have all the genes in that pathway or none of the genes in that pathway, meaning that these genes in the pathway, they should show a similar behavior, a similar expression level change based on different disease conditions or environmental conditions. So we can also, we can also ask questions like this: can we group the genes into meaningful subclasses? For example, genes that have increased transcription as the time goes on, or decreased transcription, or genes that have high expression and unaffected in these time series experiments. So we may want to group genes. We may also want to group experiments. Each of these experiments, each of these microarray chips may correspond to, for example, a single person, and we may want to find the classes of healthy people and ill people. Okay, we may need to group experiments as well, or these experiments may mean other things as well, and we may want to group them for other purposes. And classification questions are related to diagnostic purposes. For example, given the microarrays maybe in 5 or 10 years, maybe they are going to be replaced probably by other technologies, but they have the potential to be used for diagnostic purposes. For example, certain genes, if you have certain marker genes, even you don't need to, because microarrays assay, I mean, they look for expression levels for all the genes. So for a person who comes to a hospital, tries to find out which disease I have, you can look at the gene expression, if it's a genetic disease or if it's a disease that affects the gene expression, of course. You can look at the patient's gene expression level and try to classify it to other gene expression levels that you have seen in other types of diseases. Or, for example, you can understand whether it's an early, for example, if it's cancer, for example, if there are different types of cancer, you can try to predict its type by looking at the gene expression levels. And this is the classification task usually. Now I'm going to talk about clustering. We are going to focus on the clustering goals. How can we group either genes or experiments into subgroups that show similar behavior or that have something in common? So we are going to try to find natural classes, natural groups in the data. And now, in order to be, so what we have here, our data is a numerical matrix. So it's going to be a matrix like this. So our data is a matrix like this. And we have genes, gene 1, gene 2, gene 3. And if our goal is to group these genes, what we need, and each gene has a set of values, right, for these different experiments. For example, T0, T1, T2, and maybe there may be replicates and other types of experiments. There may be like tens of experiments, 60 experiments, 50 experiments, and a gene is going to have expression values for each of these experiments. Another gene also have expression values. If our goal is to group these genes, we need to be able to identify whether a certain gene, gene 4, is close to gene 1 in terms of these expression levels. So we need to be able to find out and compare these genes pairwise and be able to indicate how similar are the behavior of these two genes. We need to first do this thing pairwise. Similar to phylogenetic tree analysis. When we were building phylogenetic trees, actually we were doing hierarchical clustering. The UPGMA method that we have used, what it had these several sequences, and we were grouping these sequences based on how similar a pair of sequences is. We were given a distance matrix, and the algorithm running on this distance matrix used pairwise distances between sequences at the end to overall provide a tree, and this tree provides some groupings between these sequences. Certain groups of sequences are more similar to each other than other groups of sequences. So for clustering of gene expression data, we need something similar. We need to have a way to measure how, for a pair of genes, how similar they are. So, which brings us to distance measures. So, this gene is actually a point in a d-dimensional space. So, if I have d experiments, okay, so it's a vector, it's a point in d-dimensional space. So we can use different distance measures like the Euclidean distance. We can measure the distance between these two genes as if we are measuring it with a ruler, okay, which is going to give us the Euclidean distance. So here x and y are two different genes, and xi denotes the ith expression level in the expression level at the ith experiment. So this x is a vector for this gene. So x is this vector, y is this vector, and Euclidean distance simply, how is it computed? It's computed simply by subtracting respective columns between each other, by taking the square of these and by taking the square root of the final summation. Okay, for example, if I have 1, 2, 3, 4, 5 for one gene, and this is for the other genes, 7, 8, 1, 2, 6. Okay, they need to be same dimensional in order to be complete Euclidean distance. Euclidean distance is going to be 1 minus 7 squared plus 2 minus 8 squared plus 3 minus 1 squared plus 4 minus 2 squared plus 5 minus 6 squared and square root. Okay, so this is the Euclidean distance between these two vectors, between these 2 points. Euclidean distance is also called L2 norm. Because we are taking the square root of this. There is also another distance called L1 norm. L1 norm is simply, you don't take the square root, you don't take the squares, you just take absolute values of these differences and sum them up. Okay, this is called L1 norm. So we can define different distance measure based on how we should, this distance measure, for example, does this really biologically tell us? There's the biological questions. If you're given these two expression levels for these two genes, as a biologist, do you think these are similar or not? So any distance measure you use should be should get you to this biological relevance. A good distance measure should agree with biological similarity. And unfortunately, there is no single distance measure that you can use as the best distance measure that correlates best with biological relevance. Based on different, for example, for time series analysis, if these were time, you should take into account that this dimension, the adjacent dimensions, T2 and T3, for example, if these were all time dimension, there is a certain relationship between nearby time points. However, in this Euclidean distance, the way you compute Euclidean distance, each dimension is treated separately. If I take this 5 and 6 and put it in the beginning, you get the same Euclidean distance. If I shuffle these two at the same manner, you get the same Euclidean distance. So the order of experiments does not matter in Euclidean distance. So maybe a different distance you can devise for time series experiments, for example, could be the difference. You can take into account the difference between consecutive time points. If you take the difference between consecutive time points, if you shuffle them, you're going to get different differences. So you're not going to get the same thing if you shuffle them. So you can devise different things for different biological purposes. Pearson correlation is one of the most widely used techniques for microanalysis. Pearson correlation, it computes it's the cosine similarity. It's similar to cosine similarity, and cosine similarity is computed like this. Cosine similarity is the angle. So these are points in d-dimensional space, but these are also vectors in d-dimensional space. So points and vectors are very related. For example, in 2 dimensions, this, for example, 5.3 is a point, right? 5.3. 5.3 is also a vector. This vector is 5.3. Okay? So points and vectors are represented the same manner. So if you have 2 points, so here, to demonstrate Euclidean distance and cosine similarity, and this may be, for example, 2, 4. The Euclidean distance is the distance you measure between these 2 points as if you are measuring with a ruler. Okay, it's this distance, this scalar value. Cosine similarity is the angle between these 2 vectors. So it seems that when the angle gets larger, the distance also gets larger, right? For example, if they are vectors of same size, same length, when distance is 0, when angle is 0, distance is also going to be 0. But, so what is the difference between these 2 measures if they're measuring the same thing? There's a slight difference. In general, these expression levels, there may be certain genes which are always high expressed, and there may be certain genes which are maybe low expressed. The reason I'm saying is that the length of these vectors may be different. So here we may have situations like this. One gene, for example, 2,1, and the other is 4,2. Okay, the Euclidean distance between them is not 0. The Euclidean distance between them is 2 squared plus 1 squared, so it's square root 5. Okay, this is the Euclidean distance between them, square root 5. What about the cosine angle, cosine similarity between them? This is 1. I mean, 1 is a good thing. The Euclidean distance goes from 0 to infinity. Okay, you should also think about the ranges of these distances. The Euclidean distance is from 0 to infinity, and cosine similarity is between -1 and 1. The cosine of an angle can be either -1 and 1, and 1 means they are the cosine 0. Sorry, cosine 0 is equal to 1, which means that the vectors are pointing in the same direction. They are very similar. A cosine similarity value of 1 means that these two are very similar. And if we take the cosine similarity for these two vectors, we are going to treat them as if they are the same gene. So in other words, cosine similarity takes into account the scale changes in expression levels. If one gene has a scaled version of the expression levels of other gene, it's going to be treated as they behave perfectly similar, perfect same. But if you use the Euclidean distance, you are going to treat these two genes as if they are different genes. I mean, you are not going to treat them similar genes. So here, how you can see how distance measure is, how important distance measure is when our goal is to group these genes. Based on different measures, you can group these two genes together in the same cluster. If you use a different distance measure, these two genes may end up in different clusters. Okay, so distance measures are really important. And Pearson correlation is similar to cosine similarity, but it's computed first, it's mean-centered. We mean-center these vectors. We subtract the means of each vector from itself, and then we take the cosine similarity. Pearson correlation is used a lot for the expression, clustering gene expression data. Now, we are going to talk about our first technique. So you have understand the distance measures, right? This distance measure we need to find out how similar or how distant two genes are. Okay, we are going to we need to have a way to measure whether this gene is similar to this gene or it's different from that gene. We need to have some mathematical measure of distance. Given a distance measure, or by choosing it, after choosing a distance measure, then you need an algorithm to group these genes. For example, before talking about any clustering technique, you can directly use UPGMA for clustering purposes. So what UPGMA does is, if you can compute the distances between these genes by using Euclidean distance or Pearson correlation, you can get yourself a distance matrix, which shows the pairwise distances between all pairs of genes. Then applying the UPGMA algorithm, you are going to group first the most similar genes together. And that's a very greedy heuristic. I mean, the UPGMA algorithm, if your goal is to group genes, the most intuitive way to do is pick the most similar two genes in your data set. Out of 25,000 genes, pick the two pair which are most similar, group them together, and then pick the next similar group, either join this to this group or form a separate group. So by growing groups, what you are going to get yourself is you are going to get a clustering, a grouping of these genes. So UPGMA algorithm can be perfectly used for this clustering purpose. But is it the best way to do this grouping? I mean, it's a greedy heuristic, but can it give us a good clustering is a question. First of all, how do we evaluate clusters? If somebody gives you that, okay, I grouped these genes and these are the groups I found. These are my groups I found for these genes, for these 25,000 genes, these are the groups that I determined. If somebody gives you, if somebody else gives you another group, let me try to write it on the board. For the same data, different clusters may exist. So, for example, I have only 6 points. Okay, somebody told me that this is the way to cluster the data. And for the same data set, somebody else told me that I cluster them like this. Which one is a better clustering? Which one is a better, how do you evaluate this? Which one is a better grouping of these points? What measures? For example, what makes a good cluster? What makes a good, how would you evaluate this?
The ones with the nearest neighbors should be in the same cluster.
Very good. So one measure is what you define here is that intra-cluster distance. So for elements that are in the same cluster, the distance between them should be small. I mean, we shouldn't put, for example, I can also tell you, I mean, these both look similar to each other. They both look okay. Now, another cluster, which may use a very bad algorithm, may be something like this. Okay. Even if it will be difficult to draw this. Another cluster may be like this. Okay, this is also a cluster. Putting these two in the same cluster, these two in the same cluster, and these two in the same cluster. So in terms of intra-cluster distance, this would be performed really bad because we have put faraway points in the same cluster. Are there any measures? Are there any other measures other than the members belonging to the same cluster having smaller distances? What about this? No, not this. I'm not going. So, okay, I'm going to think about another example. For the same set of points, 6 points. Again, I'm going to make 3 clusters. Yeah, I mean, it's not demonstrating the point I want to make, but can you think of other measures than intra-cluster distances? There's a very, I mean, in terms of name, there's a very just change intra to inter cluster. What it tells you is that when you group items, groups should be far away from each other. If you decide to separate these points into 2 groups, there must be a reason for you to separate them into 2 groups. Meaning that, I mean, if you have decided to, so inter-cluster distances, if there are 2 different clusters, the distances between them should be large. Okay, I mean, if you shouldn't put close by elements. I mean, for example, this one and this one. Actually, these two, the distance between these two is smaller than the distance between these two. If you have decided to put these into different clusters, the clusters should be far away from each other. So the best grouping should group the items so that the groups should be far away from each other and the members within the groups should be as close as together. So intra-cluster distances should be small, inter-cluster distances should be large. So that's one way to evaluate clustering. So therefore, I mean, there may be many methods given, and each of them, in each examples I have here, there are all 3 clusters. But how do you know there are 3 clusters? That's another issue. What if you may also group it like this? Maybe this is a single member outside. Maybe these 5 of them, they may belong to the same group. Maybe you can group it into 2 clusters. So these are the general problems people face every day in clustering. Okay, they develop different algorithms. People still are developing, proposing heuristics, algorithms for doing clustering in difficult settings, in high dimensions, when it's difficult to separate data. So clustering is a general difficult problem that in machine learning pattern recognition. And k-means clustering is one of the most widely used algorithms. It's very simple to implement, simple to understand. It's based on heuristics and similar to expectation maximization it's an approach if you know EM. But if you don't know EM, you don't need, k-means is really simpler than the original EM. So the algorithm is just this. The whole algorithm is written on this slide. So it begins like this. We randomly assign. So let me try to run k-means on these 5 points. As the name suggests, k-means, one important disadvantage of this algorithm is that you need to know a priori how many clusters you are going to generate. The k-means algorithm cannot determine the number of clusters automatically for you. So you need to tell k-means algorithm that I want to divide my data into 5 groups or 8 groups or 2 groups. So you need to, as the user of this algorithm, you need to tell k-means, you need to give k. What is k going to be? And imagine for this example, we say divide this 6 points into 3 groups. So we give 3 is an input to the algorithm. So we randomly assign, out of these 6 points, we randomly assign 3 of them to 3 different clusters. They are going to be the cluster representatives. So we initially assign random cluster representatives. For example, say that we have these 3. I mean, I start with a bad example, bad initialization. So here in this good cluster, each of them were in the same group, but k-means may assign initially, since it's random assignment, we are doing random assignment, and by randomly, we can have this situation, having all these 3 points assigned to different clusters. Okay? It starts with assignment of 3 points to 3 different clusters. These are our initial cluster centers. And iteratively, what we do is, since these are our clusters, at the next step, we find this. Which point, this point, we look at this point, which of these 3 clusters does this point belong to? So what we do is we measure the distance of this point to the cluster center. So the cluster center are the points themselves right now. They contain single elements. So it's going to be assigned to the closest point. For example, this one. Let me name these clusters. 1, 2, 3. Okay. So this one is going to say I'm closest to cluster 1. So it is assigned to cluster 1. This one is going to say I'm also close to cluster 1. Okay. And let's assume this one is going to say I'm close to cluster 2. Sorry, 3. 2 is the longest cluster. So each of these remaining points, and we are also, if we ask the same question to these ones, obviously they are going to stay with the same. So since 1 is going to ask, I'm closest to 1. 2 is going to say, I'm closest to, because they are exactly on that point initially. So these are our clusters at the end of first iteration. Okay? At the end of first iteration, this is our cluster. Now, after all points are assigned to clusters, what we now see is that previously, this was the center of cluster 1, this was the center of cluster 2, and this was the center of cluster 3, right? This was our initial, these were our initial cluster centers. Now, we look at these clusters and see that these cluster centers are not really good representatives of their cluster centers. For example, look at this cluster 3. I mean, this point at the end of the cluster should not be a cluster center. So what we do at the next step is we update cluster centers based on its current members. So 3, the cluster center 3 comes to this point. Okay, this is the cluster center 3. And 1 comes here. 1 comes here, center of 1 comes here. 2 stays at the same position because there's only one member. Now, at the second iteration, we ask the questions again to each point. To all of these 6 points, we ask this question: which cluster do you belong to? By measuring the distances to these cluster centers. For example, this one now, this one is going to now say, oh, I'm actually close. So this one, the cluster center 1, drifted away from this point 1. Now this one, this point here at the next step is now closer to this cluster center 2 actually. So what will happen is that by measuring, by comparing to these distances, this one, and this one, it's going to say I'm closer to cluster 2. I mean, slightly closer. I mean, I don't know if it's actually, it seems closer, right? I mean, it's very slightly. You can find a better example to show that it's obvious, but here let's assume that 1 is closer to this 2. Now what will happen is that now I became a member of cluster 2. It now says at the second iteration, because these cluster centers change, it becomes a cluster center. And let's say these stay in their current places like this. Okay. Now it's going to be like this, and the cluster center is updated. This is 2. 1 is going to get farther from here. So probably at the end of the second iteration, we are going to converge. If no membership changes after we update, so this 1 moved here, this 2 moved here, 3 stayed the same. After changing the cluster centers, at the next iteration, if all members are satisfied with their current clusters, they remain at the same clusters, we stop, we converge to some cluster, we stop k-means there. And this is the final cluster we produce. Okay, the algorithm is very simple. It's iteratively, we update centers, we find new memberships, update centers, find new memberships, and in terms of running time, if we have k clusters, at each iteration we compute this many distances. For each endpoints, we compute their distances to k clusters. So for at each iteration, we are going to perform this many distance computations. And how many iterations you are going to have really depends on the layout of the points. So we cannot really tell about the convergence, and the running time is based on the iteration actually. Now, I'm going to show you an applet, very okay, make clustering. So here, let's try to do the 3 clusters. So I'm randomly assigning points. For example, let's have some cluster here, another cluster here, and maybe another cluster somewhere here. Okay, so here's my data. I generated this data for the purpose of naturally there's this cluster, and this cluster. And let's see whether k-means is going to be able to find this. I have 47 points. Okay, I have this 47 points. Now, when we say initialize, these 47 points, it's randomly. So I need to actually start. So it's randomly assigned these. Let me reset it. So let's refresh. Yeah. So some data here, some other data here, and there may be some overlaps. And there's so this time we have 65 points. Now I should be able to, yeah, I need to sell 3 here. So there's something wrong with this applet. I need to be able to start it. Clusters, 3 clusters. 3 clusters. When I say initialize, it randomly assigns new points, including points. Okay, it can insert manually. Oh, okay, I see. Oh, okay. Now I see what I mean. So we should tell, so it's like this. When I say initialize, we can actually manually insert it after we randomly initialize it. So I'm going to have my cluster. Again, I'm going to generate my points, some of them here, some of them here, and a small group of dense points here. So I have 3 clusters like this, a group here, another group here, and a more dispersed group here. Okay, so when I say start, actually, as you can see, it did, sorry for the quality of the colors, what it did was some of this group, part of this group was separated. I was expecting, so I generated points so that they should be in the same group, but the current initialization, just based on this initial cluster locations, they assigned these yellow ones into a separate cluster than these ones. And what I wanted this group to be in the same cluster, but it belonged to this larger cluster. So initially, our cluster is really bad. I mean, there's a single cluster which contains members from 3 groups I first intended. From this group, and this group. And this group only has 3 members or 4 members separated into this last group. So we can go step by step. At the end of the first step, this is the cluster centers are found based on this. And second step, membership changes. This green group actually grew. The yellow group also grew, but we still have some, we couldn't find this small group yet. Second iteration, I think it converged, yeah. So as you can see, it was able to do good, but not really good. It couldn't find this small group of points that were really close to each other. Some members from other groups are also put into this cluster. But it was able to improve the initial cluster. In the initial cluster, we had this first group split into 2 by half, but now it grew, and this yellow cluster is almost, except this single point, which is clustered incorrectly, it's almost correct. Okay, and this one, the green one, except these ones at the border, we have also found the green cluster correctly. But these ones that are at the border, they're classified incorrectly to belong to this cluster here. So it shows that k-means cannot always correctly find the good. If we use a different initialization, maybe we could have found this cluster. If, for example, our initial point, one of our initial clusters was somewhere here, probably we would be able to find that small cluster there. So initialization is really important. What people really practically do is they run k-means by like 5 or 6 times or 10 times with different initial clusters and pick the one which gives you the best clusters. Okay, so this is k-means. I'm going to stop here today, and we are going to continue with self-organizing maps. I'm going to skip hierarchical clustering. You already know hierarchical clustering. I'm going to talk about self-organizing maps on Friday. We don't have class on Thursday. It's 19th of May. It's official holiday, I guess, 19th of May. So we are not going to have a class next Thursday. Next Friday, we are going to talk about self-organizing maps, which is also another clustering technique. And then we are going to continue with biological networks after we finish this.